to be using Zoom to its full extent. We're actually not doing a two-way. It's not just London, Australia, but London, Australia, Sri Lanka, because Malika's in Sri Lanka. So it's wonderful what you can do with this thing. Before we get started, I just wanted to welcome you to this Central Association. It gives me great pleasure to hand over to our speakers for tonight. I think this is going to be in three parts. Alan is going to lead off, giving some of the background to their work, and then Malika is going to describe the work with the polytunnels and so on, and then Alan is going to finish off again. Questions and answers. If there's anything burning that needs to be cleared up at the end of each session, we'll take it then. Otherwise, perhaps we'll take questions at the end. So with no further ado, Alan, over to you.
Thanks very much, Bob. Thanks for having me. And it looks great. Actually, that Nottingham thing looks fantastic. I'm curious to read up on it. Maybe I'll see if I can come as well. So my name's Alan Dorin. I'm in Melbourne. Malika here is working with me as my PhD student at the moment. And we'll talk to you about a variety of work that we've been conducting. Some of it's like over more than a decade. Malika's stuff's in the last 2 years. And specifically focus on how we're tracking honeybees in the field using AI and computer vision techniques. So I'm pretty sure I don't really need to tell you about why we're doing this. But most of it is focused on pollination for food security. We're also very interested in biodiversity and monitoring insects in the wild. But what we'll focus on today is really about the issues associated with pollination, which isn't just big, it's really big. And obviously, when we're talking about the future population on Earth, expanding as it is, the future human population and the future of insects and other native and wild animals is not looking so rosy. And you may have seen reports of losses of insects all over the world, and papers arguing that there's been declines. There's also counters to that, and it's not such a simple story as to say there's a global decline in insects, but the loss of biodiversity is certainly going to have impact on pollination. And this is a big deal for human food security in particular, but also for food security for wild animals as well that depend on pollination of wildflowers and other crops for them to consume. So the thing that really got us thinking along the lines we'll be talking about today is the fact that, of course, the bees that we use for pollinating our crops didn't evolve to pollinate crops. And depending on the insect we're talking about, they evolved in different foraging environments. So bumblebees evolved in temperate landscapes where meadows and wildflowers are often distributed, as you can see over on the left. Whereas in a tropical landscape, often you'll find one or two trees in flower, the same species at the same time, and there could be quite a distance between those trees. And yet when you get to that flower source, it's a single source of flowers, all of the same species that might be many thousands and thousands of flowers large, rather than the dispersal that you find in a wildflower meadow or something similar. And of course, that's really not then the environment in which we ask the honeybees or bumblebees to forage for us. Now, in Australia, we've got around actually 2,000 species of native bee, most of which we know very little about. And for the most part, we don't know a lot about what they contribute to pollination of our crops either. They're everywhere. You can, if you look, you can go around and find them just in back gardens, which is quite surprising to people. So when I started doing, research into bees, my background is in computer science. And so I was introduced to this idea now more than a decade ago, that we had 2,000 species of bees. And I'd always thought of honeybees and bumblebees and never really thought much more about it, of course. And now I realize how silly that is. And there are about 20,000 species of bees worldwide, but 2,000 of them are in Australia, including these beautiful blue banded bees, you know. And when I describe to my friends, oh, there are blue and black striped bees, they're like, what? You've got to be kidding me. They're black and yellow, right? Because that kind of iconography of the black and yellow striped bee is so ingrained in our culture that all these other bees are hidden, basically, and people don't know about them. So we have all these bees that have, you know, evolved in landscapes like Australia or tropics or in temperate areas like in Europe and the UK. And then we go and dump them in the middle of fields. This is a potato field from the Netherlands. Or in a greenhouse like this. This is a massive piece of work in the desert in Australia, actually, where they're growing tomatoes on recycled water in a very high-tech facility. And, you know, this isn't the kinds of environments that the bees are used to working in. This is our strawberry farm that Malika and I have been working on for the last couple of years. And every direction you look is the same. There's plastic overhead that's interfering with illumination and scattering the sunlight. There's hundreds and hundreds of posts and hundreds of rows of strawberries. And if you put me in the middle of one of those things and spun me around with a blindfold, I guarantee I would get confused about which way to go next. And yet we insist that the bees do a good job of pollinating a crop that they don't particularly like in an environment that is particularly confusing, under conditions that are often too hot and uncomfortable for them because the polytunnels get quite warm. So we thought, well, what can we do about this? And the first thing that really matters is that we understand what's the current situation. So we started asking a number of questions. You don't need to read these in detail, just a sample for of the kinds of questions that we're asking. But in particular, in this case, what we're concerned with is how can we lay out our agricultural and horticultural infrastructure in such a way as to better enable the bees to use their innate abilities to navigate in order to improve both the situation for the bees so that they don't get so stressed and confused in these environments, and as a result to improve crop pollination. So we're looking at things like navigation facilities, temperature control, illumination so that flowers are easy to detect, for instance. And to make sense of all of this, we've got to, of course, understand what the bees are doing under different conditions that are experimental or that are just the ones that are there by default. And so that means we need to be able to monitor those insects and their behavior. So we started with this, this process, which has now become a little bit more refined. And it's not rocket science, this process, but basically we start by talking to the people on the ground who are the experts. So in this case, that's the beekeepers on a particular site and the growers on the same site. And after we've discussed really their kind of evidence for what's going on in their location, we move on to our own technologies and techniques for observing what insects are around in that area and how they're behaving in the crop. We then go on and we build computer simulations that take into account the parameters we've extracted from the data that we've monitored. And we try out different modeling scenarios to see, can we improve the situation? Our software will make some predictions about how changes we make can influence the outcomes for pollination on the crop. We'll recommend some changes based on the outcomes of our simulations to the growers. Hopefully they'll implement some of those recommendations, and then we iterate around this loop. So now I'll just go through most of these steps in a little bit more detail, and Malika will as well, in order to just kind of fill in some of these sections here in the process. So the first one relates to what insects are in what areas. Now, in different types of landscape, in different climates, and of course in different countries, we've got different insect distributions. And that's changing under climate change as well. And one of the sources of presence and absence data or occurrence data for insects that is quite useful in a country that's as large as Australia is actually social network site data or social media data. So people posting online pictures of insects that they've seen by the thousands in many cases. And we've mapped those out in a number of different case studies to explore how much we can learn about where bees are in a country as big as Australia. So for those of you who haven't been to Sydney or wherever, the whole of Europe and the UK all fits within Australia, and there's heaps of room to spare, right? That's how big this country is. So we can't send ecologists out everywhere to tell us, oh, what insects have you seen here or there? And over the last couple of 100 years, you know, certainly ecologists have been mapping where insects are found, but that's not enough if the insect populations are moving. So that's why we're looking for alternative techniques. So this article, without going into any detail, takes data, image data from a number of different social network sites, including Flickr and Facebook and Twitter, and also proper databases of species biodiversity. And it uses computer vision to scan through the images on those sites and to identify which ones are of the species that you're interested in. And then we rank them according to the number of occurrences of that particular species on that site so that we can identify which are the best sites for a particular species. And it does depend on what species you're looking at. Then once we've identified which of those websites or social media sites has the best kind of insect data for the species we're looking at, we actually do a much more detailed kind of data harvesting where we identify the species in the images, again, automatically using machine vision and deep learning techniques. So that's a sort of an AI technique that's able to look at the image content and recognize a species that you've trained it to recognize. So in this case, what you can see is my favorite little bees, the blue banded bees that I was talking about before. The green tags on the map show some of the data that's been found from, in this case, Flickr and its location. The red tags show some data that we've excluded. So when you do a search, sometimes you get things back that might not be appropriate. For instance, you might find a pinned insect specimen photographed in a museum, and you don't want to include that in your distribution charts, so we'll tag it. The yellow shows the range as was known for this species from the data within the Atlas of Living Australia, which is the kind of our node in the global biodiversity information system for species biodiversity. So what you can see from this is there are a couple of locations where the insect has been seen that weren't previously known, and that would probably, you know, suggest to us that we should send out an ecologist to investigate that location. And in a country like this, okay, you can see there's a scale here. So we're talking about 4,500 kilometres across the middle of Australia here. Obviously, it's useful to get any data you can before you start sending people out to look for where the insects are. So we can overlay maps like this onto farm sites so that we know what insects are likely to be in an area even before we start to visit that site. And then once we've identified what insects are there and we know that we've got a particular site in mind, then we can start setting up much more detailed instrumentation. And at this point, it would probably be good for me to hand over to Malika to start talking about what he's been doing. Malika, just call out to me when you want me to change the slides, and I'll do that. Okay,
sure,
off you go, Malika.
Yeah, thank you very much, Charan, and thank you very much again for having us. So let's, I'll go through this slide first. So as Alan mentioned before, to understand the impact of insects towards pollination and their effectiveness, we have to identify what are the insect types there are, how many insects are there, and what they are doing in setups like agricultural farms or horticultural setups. So this is usually achieved through insect monitoring and surveying. So I have integrated several current insect monitoring techniques here. So I will briefly go through each of these and discuss their advantages and disadvantages. So the first one is pan trapping. So this is also called as ball trapping. So in this method, ecologists will fill colored balls with soap water and leave it in field for few days to insect to be collected. So later the collected insects are taxonomically categorized and counted to predict the impact of different insect on pollination. So the sticky traps and vane traps indicated here follow the same procedure more or less. So in a sticky trap, instead of using balls, we use colored papers covered with glue where insects are being trapped. So here you can see a sticky trap. So those are passive methods of insect data collection, and we have the active methods as well, which are shown in below. So one is the direct observations or contact observation. So here usually trained ecologists observe a specific area to record and count all the insects visiting that area and record their foraging activity, what they are doing, which flowers are they visiting, and so on. And the next one with sweep netting technique, ecologists use nets shown as here to catch insects to count and identify their species. So as I mentioned before, pan trapping, sticky traps and vane traps are considered as passive methods of insect monitoring, while the direct observations and sweep netting are more of active methods. So these are some of the methods which are widely used currently, and there are some methods in addition to this. So let's see the advantages and disadvantages of these techniques and how computer vision and artificial intelligence or AI can be used to overcome the disadvantages and automate this insect monitoring process. So here I have listed two traditional insect monitoring techniques which we discussed earlier and compared it with computer vision facilitated automated insect monitoring. So here I will refer to this direct observations and pan trapping as a traditional method because we have been using it for a long time. So first, if we consider the difficulty of use associated with this direct observations and pan trapping, the difficulty of use is high for these methods as they require researchers with high expertise to conduct accurately. However, with computer vision based techniques, we can train neural network to detect and track insect automatically. So next, when it comes to the source preservation or keeping the records, direct observations cannot preserve the resource of the original recording, but pan traps and computer vision based techniques do preserve the recording. So both of these traditional methods are prone to biases related to the observer or the researcher. However, with computer vision, since we are using a neural network or computational methods, they are usually not prone to biases. Next, if we consider pollination, flower witches are very important for pollination or recording pollination. Pan traps provide no functional evidence that an insect does visit or pollinate flowers. But with direct observations and computer vision, we can monitor and record flower visits. And finally, most importantly, actually, data collection and processing require manual interference for traditional insect monitoring methods. But through help of computer vision, we can automate this process and deploy it in a wider range or wider area. So as you can see through these points, computer vision and AI facilitated insect monitoring can overcome lots of drawbacks associated with traditional techniques. So next slide, please. Thank you. So let's see how this computer vision and AI can be used for insect monitoring. For this, our research group, we propose three-step process shown here. I will explain the component of this process in coming slide, but first briefly I'll give an overview of this process. The first step of this process is the video data collection. Here we collect video data of insect foraging using portable cameras installed in the field. So you can see some photos of cameras in the field, and in the below, that's a weather station. We can link our video data or insect foraging data with weather data and see how the insects counts vary based on the weather as well. Next, in the second step, these videos are processed automatically using computer vision and AI algorithms to identify flower positions, detect insects, identify their species or type, and track their movement through space. So this is the actually the stage we are using our algorithms to track insects. And finally, in the third stage, insect movements, flower positions, and other data are further analyzed to infer on pollination, make predictions, and evaluate pollination effectiveness of each insect species. So this is an overview. In the coming slides, I'll get into the fine details related to these three processes. And say thank you. So first we'll look at the video data collection. So as I mentioned before, here we use cameras installed on fields to record insect activity. On the left side, you can see a Raspberry Pi based camera deployed in a strawberry field. So Raspberry Pi is a small edge computing device which also can be connected with a camera. So here it's deployed in a strawberry farm, monitoring and recording insects visit to strawberry flowers. And on the right side of this side, you can see a snapshot of video recorded from one of these cameras. The area covered by the camera usually depends on the resolution of the sensor and the frame rate. So a high resolution camera can cover wider area, and as well as a high frame rate camera also can capture the insect or very high speed insects like wasp and blue banded bees. Next slide, please. Thank you.
This is so good.
So here we look at the automated video processing stage. Here recorded videos which we recorded in the previous stage are transferred to a computer for processing or a processing device. First, the videos are dissected to individual frames for processing. And on the each individual frame, we run computer vision and artificial intelligence algorithms to detect insects if there's any. So in this figure in the middle, you can see a honeybee detected using computer vision algorithms. After insect is detected, the AI algorithm, which is usually trained with thousands of insect images, identifies the species or the type of the insect. This helps to later compare the pollination efficiency and efficacy of insect species. Next, in the bottom right, the insect is tracked in subsequent frames to create a track of the insect movements. And finally, insect movement tracks are superimposed to generate a map of insect movements as shown in the figure in the bottom. So from this, you can see what are the most favorite areas or areas for insects and which flowers are visited most by insects. Next slide, please. So now I will describe briefly describe the technical background of the tracking process to those who are interested. It is not like critical to understand the details, but I will explain this in summary to give a better understanding of the working mechanism of our algorithm. So I first explained about the video data collection in the left hand side. So next I will explain the automated video processing in our methodology. So to detect and track insects, we use a hybrid detection model when we are processing the video. So as the name suggests, the hybrid detection model has two detection models. One is the AI-based method, which is computationally heavy, and other one is a computationally lighter method. So the heavy one is a AI-based method based on deep learning. It can detect the position of an insect and identify its species in a given frame, and is very comparatively very accurate. The next method, the light method, is called the foreground and background subtraction. So this is an old method which has been used for years, and it works in most circumstances. But if there are like constant changes in the environment, such as wind or changes in the sunlight, this does not work accurately. Also, this cannot accurately identify species of insects. So we use one of these methods to detect insects in the video depending on the circumstances, and the algorithm, our algorithm, hybrid detection algorithm, intelligently switches between these two detection models to detect insect and track insects. And also, when possible, we process the video in low resolution because it increases the processing speed. But sometimes that is not possible, so we have to switch to high resolution processing, which might give the information that we require, but it will drain on computational resources. So after we use the model to work out where the insect is, then we have to like link up from the position of insect from frame to frame to create a track. So we do this using prediction model called constant velocity model and a data association algorithm called Hungarian algorithm. So if you have any questions related to these two methods, please let me know. So we use this process to generate insect tracks and their flower visitation data, which is used for pollination analysis. Next slide, please. So here we have a video of insect being tracked in wildflowers. Actually, here we are tracking honeybees, and the algorithm will identify when honeybee goes under the foliage and return back. So in this version of the algorithm, actually we were tracking one honeybee at a time, and later we extended it to track multiple insects simultaneously. We can see a video of that in the next slide after this video. So this is honeybee being tracked in strawberry flowers. So as you can see, our algorithm can track honeybees amidst the changes in the environment, like as wind conditions. Okay, so so that's about the algorithm and our method. So next, let's look at the case study we conducted in strawberry farm using our algorithms and monitoring techniques. So the main objective of this case study was to monitor and assess the pollination across a large-scale strawberry farm. So we looked to achieve this main objective by counting pollinators and comparing the contribution of different insect species to crop pollination. So here we observe the contribution of 4 main insect types, which were being observed to be foraging in the strawberry farm. So they were honeybees, syrphid flies, mainly hoverflies, Lepidoptera consisting of moths and butterflies, and wasps. Next slide, please. So we conducted our experiment at Sunny Ridge Strawberry Farm located in Victoria, Australia. It's the same farm actually Alan mentioned. In the bottom, you can see a map of the strawberry farm. And Sunny Ridge mainly grows strawberries in protected polytunnel. So we monitored the honeybee or insect pollination activity in these polytunnels. We installed camera units in 9 study locations to cover the edges and the middle of the polytunnels. And a map of these locations are here, shown in the right of the screen. Next slide, please. So we recorded videos using camera units consisting of a Raspberry Pi computer, which I mentioned before. Each camera covered an area of 60 centimeters by 40 centimeters. So a snapshot of this video taken from a camera is shown in the bottom right. So the data collection period was from 11 a.m. to 3:30 p.m. for 6 days. And next, I will present a brief overview of the results. Thank you. So here I have shown the results recorded from these cameras in the data collection point 1, which is shown in the map here. Here we recorded over 340 insect tracks and 45 flowers over 6 days period. In this chart, the top 2 bar charts show the number of tracks recorded and the number flower visits recorded broken down based on the insect types. And the map in the bottom shows the insect trajectories. So majority of insects recorded were honeybees, and there were also few wasps, but there were hardly any syrphid flies or lepidoptera. Next, if you look at the flower visitation data given in the second bar chart, most flower visits were made by honeybees, indicating that they were the main driver of pollination. So this is the data we collected from one location. And in the next slides, we can see the data recorded from all the locations over 6 days. So this is maps of all the data recorded from 9 locations over the 6 days. So we recorded over 2,300 insect tracks in total. So this fine scale data help us to identify the pollination efficiency and efficacy of each insect species in the farm. Also, we can analyze data further to predict the crop yield and assist growers in better designing agricultural spaces. For example, we can use data to identify the most optimum areas to place beehives to maximize pollination levels. So next, Alan will describe how we use data in simulations and to better inform pollinators. Thank you.
Thanks, Malika. Okay, so we collect all this data, which, as you can see, is a lot of data. And then the question is, well, what do we do with it? And of course, there's if you like, the standard approach. You look at the data, you draw conclusions from the data, and then you jump straight to your predictions about what you could do to change the behavior. So for instance, if there weren't enough honeybees on the crop, we could just go to the beekeepers and say, okay, can you please bring in some more hives? But as Malika mentioned, it could be more complex than that. You might want to know, well, where should you put the hives? Again, you could look at the data and say, well, at location 1 in this polytunnel, there weren't many bees. So let's put the hives over near location 1. However, bees, as I'm sure you know, don't always behave in the way you would like them to behave. And on this farm in particular, for instance, we know for a fact that the strawberry growers pay a lot of money to have hives placed on their farm verges. And we also know for a fact that many of those bees go to the next door farm. So the beekeeper is paying all this money for the hives, and the bees are happily pollinating the crop next door. And it doesn't really matter where you put the bees. They actually honeybees don't like the strawberries very much, which is also something that we've been able to establish with our monitoring. There are weeds on that site, which the honeybees absolutely love. There are raspberries on that site, which the honeybees also like. So the question is, well, what can you do that's better than just dumping more bees or moving bees randomly around the site? And this is where computer simulation comes in. So what we're doing with this is basically building a virtual bee. And you can think of it like your favorite computer game character. I don't know if any of you have seen even a game like, say, from the 70s or 80s, like Pac-Man, where there's a little creature moving around a maze, and that creature is being chased by ghosts. The ghosts are all under computer control. Now, what we're doing here is building a simulation a bit like Pac-Man, but there are no ghosts and there's no little creature eating power dots. There are bees moving around under computer control in a maze, which is the field site that we're interested in understanding. And those little agents, we call them, or bee agents or virtual bees, have their own vision systems, their own control algorithms, their own interpretations of colors, decision-making and learning capabilities, all based on the data that we collect in the field and what's known about experimental performance of these insects in the lab. So we build up a better and better picture about how the bee behaves, what it sees, how it responds to what it sees. And from that, we come up with a complex bee agent. Then the next step after we've got this little bee agent is to let it loose in a different foraging environment. Over on I'll start on the right just for simplicity, is a two-dimensional, you can imagine a carpet of flowers, in this case of two species, labeled here as red and blue dots. That's showing a field with intermingled flowers. And then you can see some little pink squares there. They're locations that have been visited by the bees, and pollen has been carried from one flower by the bee's body to the next flower and deposited on the stigma of this virtual flower to pollinate it. And then based on these movement patterns, of course, we generate pollination behavior. On the left, you can see a three-dimensional model of this. So in some crops, the flowers aren't laid out in an even carpet. So for instance, tomato crops, the flowers are part of the vine structure, which extends quite a way vertically. So the bees can actually have to navigate through those complex three-dimensional structures. And in that case, it's very important to build a three-dimensional model that takes account of things like occlusion, so hiding flowers behind leaves and so on, which doesn't happen when you've got a crop that's predominantly a carpet of flowers. Now, unfortunately for everyone, three-dimensional computer graphics and simulation is a lot more computationally expensive than the 2D one. So to do the kind of model you see on the left, we had to use a supercomputer. To do the one on the right, we do still use a supercomputer because it's fast, but you could also run it on your laptop, even if it's a bit slow. So this is just two examples, and you can see the paths of the bees in each cases, and you can work out from that. On the left, you can see the pink flowers are the ones that have received pollen from another flower and have been pollinated. And that enables us to make predictions about under what circumstances a crop will be pollinated. And we know that people plant their crops in different ways, and that's one of the things we're looking to understand. So for instance, in a community garden, there's one on the left there from down the road from me in Melbourne. People get very small plots of land. Maybe they're 2 meters by 3 meters across. And we were interested in understanding if everyone planted a different vegetable needing pollination on their plot, what does that mean for pollination, given what we understand about how honeybees prefer to keep visiting the same plant or the same species again and again? So we did some simple simulations laying out plots of land. You can kind of see a rough approximation of the world here with 2 different species in this case, just on a sort of a checkerboard or not like a chessboard, but like alternating plots and randomly arranged plots. And then we investigated the time to pollinate that crop based on the number of visits that the bees would make to each location based on their own internal conditions. And so you can see we end up with this curve where we identified the kind of optimal plot size somewhere around 11 meters for, in this case, the honeybee crop. Now, Yeah, honeybee pollinated crop. If you go much smaller than that, down to a plot size of 1.4 meters, you can see that pollination was very slow. If you went very large, like industrial agriculture, you certainly get a greater rate of pollination, but it's not significantly bigger than, even just 11 meters. This curve's quite gradual. Again, I'm not going to go into the detail about this. You can ask questions about it later if you want. The point is just generally to show you how we use these simulations to understand what the implications are for different agricultural setups. So here's another thing that you've probably come across before. When you're planting seed crops, your aim is to get as many pollinated female lines as possible. So you want to minimize the number of male lines that are the pollen donors in your greenhouse. Now, if you make it too small, then you don't get adequate pollination of the female lines. If you make it too large, that is, you've got too many male lines, you're wasting space that you can't sell the male like seed. There's nothing fertilized there. So that space that could have been occupied by a line of females that were being pollinated, producing seed that you then sell. So the aim is to minimize the number of male pollen donor lines in your crop and maximize the number of females. But how do you know how many is too many and how many is not enough? So we ran simulations to work out where you should put the male lines and where you should put the female lines, and comparing different numbers of male and female lines for this hybrid seed production in greenhouses. And you can see on the graph over here, this was the industry planting arrangement that was currently in use at the site where we were running these trials. And we potentially discovered a 71% increase in yield by changing the number of rows and their arrangement to kind of capitalize on the way the insects were moving between male and female lines in the crop. Unfortunately, we didn't get to complete that project, so we haven't actually tested the outcome of this one in the field, which is a bit disappointing, but sometimes industry partnerships wind up sooner than you would like. This one compares the visual system of bumblebees and honeybees. As I mentioned at the start of the talk, they actually evolved in different environments. And because of that, they respond differently to the presence of distractors. So a distractor is a non-target flower. So if you imagine you've got a flower crop and there are weeds growing through it, actually, you can see here that bumblebees that grew up in environments like that, when I say grew up, I mean they evolved in environments like that, their performance is quite constant. Whereas honeybee performance as pollinators declines, the more distractors are present in the environment. So the more there are weeds or other crops in the environment, you see this sudden drop off. This is just looking at how we understand flower color from the perspective of a bee. So our model doesn't take into account human vision. That would be a bit silly. Bees don't see the world the way humans do. So we have to accurately measure the spectral reflectance of the flowers that we're interested in studying. We do that with a device, a spectrophotometer that's specifically set up for that purpose. And it goes down into the UV range where the bees can see. And then from those measurements, we generate a reflectance curve that shows us how light is at different wavelengths is reflected from a surface such as a leaf or a flower petal. And that enables our bee vision model to accurately reflect what a bee can see and what it can't see in a particular set of illumination conditions. And that's part of the input to our model. Here's just some results from some of the experiments we did do by inserting what we call bee baffles into polytunnels. So the bees tend to move along a row when they get into a polytunnel. Now, if you've got your male and female lines or your single species lines planted in rows, that's not very good for cross-pollination. So what we did at various intervals is insert a flute board, like a cardboard panel, just in the way of the bees to force them to go along a row and then divert across to the next row or to fly over it or do something else. And we placed those baffles based on the locations that our simulations said would be the most effective locations for the baffles to improve pollination. You can see some of the trial results. So the experiment here for the celery seed production improved the yield by 127%, so more than doubled the crop yield compared to the control. Red cabbage, we got a 93% improvement. Again, in another red cabbage tunnel, we got 58% improvement. We had some issues with infestations of local pests and fungi and all kinds of problems with some of the crops, which made it very hard to understand the results, but in both cases, we didn't get a very strong result. This one, we had a minor issue, but in general, it was thought that the data was reliable. So you can still see a 5% gain there. So that's really the kind of simulations that we're doing. A variety, as you can see, there are others underway and some not yet published. Malika and I are happy to take questions from you. And we hope you found the talk interesting. And thanks very much for your attention. You're muted, Bob. Bob, you're muted and we can't hear you. Ah, that's it.
Got me. Right. Yes, sorry about that. Well, Alan, you've disappeared from my, where are we? Add spotlight. Good, right. Fabulous. Thank you very much. Yeah, that's really interesting. Amazing what you can do. And some of those yield uplifts were quite remarkable. Well done. We have got a few questions coming through, of course. Can I get one in first? I live in Kent, which is sometimes called the Garden of England. There's an awful lot of polytunnels around me and a lot of strawberries. And the pollinating insects they use are bumblebees, not honeybees. You have bumblebees. I was just curious as to why honeybees.
We don't. No, honeybees were introduced here 1830s or so, very early on, like more than 100 years, but not bumblebees on the mainland.
Oh, okay.
They were introduced illegally to Tasmania in the last 20 or so years, probably from New Zealand though. And it's suspected by tomato growers because the buzz pollination that the tomatoes require obviously can be done by bumblebees, but not honeybees.
Yes.
So the blue banded bee that I was showing, it's on the screen now, actually, that is a good buzz pollinator.
Oh, okay.
But they're pretty hard to look after. Some people have been exploring pollination with them in tomato crops, though, including in enclosed greenhouses. So no, we don't have bumblebees.
Oh, okay.
Not on the mainland.
Right. Okay, understood. Right. I don't know if people who've asked questions would like to unmute and ask them themselves. Gino has a couple of questions. Gino, would you like to unmute and ask your questions?
Sure. I've just found the button. Can you hear me?
Yeah.
Yes, thank you.
Yeah, so very interesting work, guys. I was interested, how reliable is your identification algorithm? And what features is your deep learning algorithm latching onto to actually do identification as well?
Malika?
Yeah, our algorithm, so it's the reliability mainly depends on the number of images we use. So currently it's over around like 82, the reliability depends on the insect type. So we have used a lot of images of honeybees. So in tracking honeybees, it's very reliable. But when it comes to for like insects like wasps, because of their speed, it's not so reliable. But currently we are looking at how to differentiate insects based on their movement or based on their behavior, which will further improve the reliability of our method. And for the second question, for the features, actually we are using an algorithm called YOLO, a deep learning algorithm called YOLO, which to detect insects or as the AI algorithm. So in deep learning, the algorithm actually like identify the features to use for tracking for detection by itself. So we are not specifying any features. We are only using the image of the insect to actually train the algorithm with. Hope I answered your question.
Thank you.
I'll have to think about it. May I ask one more question before moving on?
Sure.
Now, it's quite fundamental. So when you detect an insect on a flower, I believe there's a fundamental assumption that pollination has taken place.
Yeah.
But that might not be true. Have you done any clarification on that?
We have actually, and we're looking at extending the algorithm. So Malika's software will pick up even the location. So it identifies the location of the flower. At the moment, we just draw a circle around the flower, and any stop on that location counts as a pollination potential anyway. But we're actually looking at being able to detect the presence of pollen, so deposition onto the body of the insect in order to better ascertain whether or not it's, pollination event. And then I do have another student who's looking also to identify the subparts of the flower. So you'd be able to find the stigma, for instance, and then determine whether or not pollen's been, picked up from the anthers onto the body and deposited on the stigma as well. So we're certainly looking into that.
Great, thank you so much.
Thank you. Pam Hunter has a question. Do you like to unmute, Pam? Are you there? Technical problem, probably. Okay. She asks whether you could repeat the bit about the distraction of honeybees by weeds, and just go over that one more time.
Sure. So I'm just going to flick back to the slide quickly.
I stopped sharing.
Yeah. So basically, the honeybees and bumblebees scan their visual environment in a different way. So bumblebees more or less take a snapshot of their environment, and in a constant amount of time can identify their target flower species. So let's say they're interested in a species of flower that's, I don't know, just for simplicity, blue, and they'll look at this scene before them, and they'll identify in a constant amount of time where the next blue flowers are. It sort of pops out at them. That's my target. Honeybees, on the other hand, have to scan their environment to identify where the blue flowers are. Now, what that means is that for bumblebees, when you introduce a range of other flowers as well as the blue flowers continue just to pop out at them, and they can see them in a constant amount of time, regardless of how many other flowers, non-target flowers, the ones we call distractors, are present in the environment. And that constant amount of time, obviously you can see there, is visible because the graph's horizontal, right? So it doesn't matter how far you go along the horizontal axis, increasing the number of weeds or other flowers, the time it takes the bumblebee to spot the the target, is constant. Now, the honeybees, in the case where there are no distractors, is faster than the bumblebee, because the honeybee evolved in an environment where you might have a big flowering tree where all flowers are the same. So they're not actually used to looking for a flower amongst a lot of other flowers. And when I say used to, we're now talking about their innate capabilities. Bees all learn, and that can change things, okay? But this is the kind of simple scenario. But then as we increase the number of distractors in an environment, so that the honeybee has a flower that it's looking for, and there are more and more other flowers that are the distractors, right? The target becomes harder and harder to spot amongst the noise of other flowers. And so the performance, the rate at which the honeybee is able to spot its target decreases quite rapidly. And that's been shown in empirical, like in experiments done in the lab. So we took that data, built our computer model, and then tested out some implications. Does that answer the question, do you think?
Pam, are you happy with that? Yes, I saw you nod. Right, lovely. That's
Thanks,
good.
Pam.
Thank you. Sophia, would you care to ask your question? Again, maybe not. Okay. This was asking why you used Raspberry Pi camera and why was the programming of the camera important?
Malika.
Yeah, so main reason we are using Raspberry Pi camera is actually the thing you said, because we can program it accordingly based on the environment and based on the insect we are going to track, and also it's comparatively cheaper to compared to other options. And and one more thing is like for in future currently we are recording all the videos of insects continuously, and then we are processing those videos. But with Raspberry Pi in future we can actually do some kind of pre-processing in the camera itself, where we only record the videos when there are insect presence. Insect is there, so that will ease the burden of post-processing. And so that's the second reason we are using the Raspberry Pi camera.
Yeah, so we can use the Raspberry Pi. It's a computer, so we can use it like a camera trap that's programmable. So we can turn it on and off when we want to. We can make it detect when an insect moves and then start recording video and stop after a certain time. We can link the data on the camera to other sensor data. So the weather sensor data that Malika was talking about can be associated with an individual Raspberry Pi. So we can therefore tell what's the temperature at this particular location in the polytunnel, as opposed to one on the other side of the polytunnel, which could be several degrees different.
Yeah, okay, that's good. Robert Pickard has a question.
Great talk, really enjoyed it. If you're in horticulture and you're trying to cross different varieties of the same species of flower, sometimes the bees will go to one but not to the other, so you don't get cross-fertilization. So I just wondered if you shine some dominant wavelength of light over a mixture of hybrid flowers, would that stop the bees discriminating between them and dramatically increase the cross-fertilization of the varieties?
You could try that. So basically, I think what you're saying is you want the two flowers to look the same to
the bee.
The insect. The thing is that the insects are they're smart, and so you might say, well, okay, here are two species of flower, and you would collect their curves. I'm not sharing my screen at the moment, am I? You would select their curves, and then you would try and compensate for the drops in one curve with the dips in the other to sort of even out the spectra so that visually they look the same. Look, it might work in theory. I don't know if it would work in practice, but certainly, like for a fact, I can say that yes, changing the illumination will change the way that the insects perceive the flower petals. So you could definitely correct for things like drops and peaks. But how would you'd have to isolate the lighting, and spatial location is one of those things that bees are quite good at understanding. So if there was some flowers under one illumination and then flowers in a different location under different illumination, two things are going to happen. One is the bees will know that location A is not location B, and therefore they'll be treated differently. And the second thing that's going to happen is they may actually learn, for some other reason that you haven't thought of, to prefer one over the other. And their visual systems, just like ours, will correct for changes in illumination. Yeah. So you've seen that if you look at a white sheet of paper at sunset and you look at the same sheet of paper under the noon sun, and then you look at the same sheet of paper at noon but in the shade under a tree, the light reflected off that surface is different each time. But you just see it as a white sheet of paper, because your brain is correcting for the changes in illumination.
Yeah.
And the bees do that too. They've got little sensors basically on the back of their head that give them an indication of the current illumination, and they use that to adjust their, not consciously, it's part of their visual system, to adjust and color correct, if you like.
Right.
Good question. I would love to try that, but I don't know if it would work or not. It's really interesting thought, Robert.
Okay, well, we don't seem to have any more questions. I'm surprised. There was some fascinating talk. Thank you very much indeed. It's just wonderful the way that you know, science can be applied to these really complex problems, and I can see the huge potential of you know, artificial intelligence in these situations. Yeah, absolutely wonderful. Well, so thank you both. Thank you to Alan for getting up early, and to Malika for talking to us from the middle of the night. And thanks everyone for joining us this evening for a really interesting talk. So thank you very much again to everyone, and cheerio.